The genome is diverse. It takes diverse forms, as diverse as the different propagating entities, including cellular and acellular life forms or propagating entities are. They differ not only in the shape, such as whether they are circular or linear, or not just in the numbers, if they are single or multiple and how many. They also have diversity in the way they are organized in the genome and the constituents and what all they encode as well. So one can say that complexity is proportional with that of the genome, or the cellular complexity increases with the genome. We'll try to understand more of these as we progress now. First things to define is something like the open reading frame. The whole genome, especially the ones that are coded as proteins, or the products of the genes or proteins, then the language is the basic component in terms of genetic information becomes a codon. A codon is a set of 3 continuous bases, something like this. If there is a continuous base, then we are in a codon consists of 3 bases. And say, for example, if you are counting this, it would have been a codon. If you're counting these 3 bases, TGG, that would have been a codon, and ACT would have been a codon. And that is the next level of understanding is if we put all these 3 codons in a line or consecutively without any spaces or addition of any other base in between, that is a reading frame. And if that reading frame starts with a start codon, that is, here it is written as AUG, it could have been ATG if you are talking in terms of DNA, AUG if we are talking in terms of RNA. So an open reading frame, which is abbreviated as ORF, is a stretch of codons starting with a start codon and consecutive. There are multiple consecutive codons and end with a stop codon. The stop codons are TGA, TAA, and TAG. So if a stretch of DNA is given to something like this, and then we should be able to identify the open reading frames. So the first thing we look at is trying to see if there is any ATG, because every open reading frame or ORF should start with ATG. This is a DNA sequence, so therefore we'll look at ATG as well only. So as the answer is given here, ATG, this is the start codon, and we have consecutive codons. We are reading them as they are, something like this, until we come across a TGA, and that is stop codon. So this small stretch is an example of an open reading frame. Typically the open reading frames will be larger than this, the ones that are coding for protein. For illustration purpose, I've made up a sequence. Not just because we find one open reading frame, and that doesn't mean that's the end of it. There could be multiple other open reading frames too in the same stretch. Say for example, another one, ATG, is starting here in this reading frame, ATG, ACA, CAT, and TAG, that's a stop codon. So this small stretch could be another open reading frame. Not only that we would find it in this direction, on the top strand, it could be also on the bottom strand. One thing that we always have to remember is all the codons, all the open reading frames should always be read from the 5' to that of 3'. So if you look at the complementary strand, we can see that there is a 5'. It is anti-parallel to the top strand. That is 5' to 3'. And if we look at for ATG and open reading frame, we can see this ATG, TGT, CAT, AAG, GTC, and TAG, which is a stop codon. So we have in this small stretch of DNA, we have 3 open reading frames. These two open reading frames, open reading frame 1 and open reading frame 2, are overlapping. One open reading frame is a part of another open reading frame. Some of them can be even more overlapping as well. So what we can also discuss is, this is called as gene density. Gene density is because that means if you have about 1 MB of DNA, how many genes are present in it? If you take majority of the viruses, they have very small genes, but they can code for multiple proteins, and that is because they have these overlapping open reading frames, and sometimes open reading frames on both these strands. And that is why the gene density of viruses is much more higher compared to that of other organisms with more complex lifestyles or complex abilities. Viruses are very complex in that, but they are parasites. So if you look at different something like gene density and number of introns and repetitive DNA, as the genomes increase in complexity and size, we will only find that they have more introns and repetitive DNA. Let's just compare the cellular life forms, that is E. coli, Drosophila, humans, and Arabidopsis. The genes per MB, which is indicative of gene density, is highest with 954 bacteria or E. coli, represented by E. coli, then Drosophila 82, humans 62, and Arabidopsis thaliana 220. There are several other plants which have gigantic or polyploidy. Because of polyploidy, they have gigantic genomes, and their gene density could be high as well. Not that all the genes would be expressed and at all times. It is always differential expression. The important things of why gene density is less, if you look at humans, for example, compared to that of E. coli, is we have large amounts of DNA that is something like introns or junk, or we don't yet know the value of it at this time. So the E. coli doesn't have any introns, so it doesn't need much of a splicing mechanisms, whereas Drosophila has about 3 introns per gene on average, and humans have about 6 introns, and Arabidopsis thaliana has about 3. And percent repetitive DNA in the whole of genome, if you look at E. coli, has very less of it, whereas 2% of Drosophila could be repetitive DNA, whereas humans and Arabidopsis thaliana is about more than 40% of them, of the genomes, is repetitive DNA, which is astonishing, and we don't know if it is really useful or if there is any reason for having these kinds of this much of repetitive DNA. So I hope we could recollect the open reading frame. That's an important one, which will, because we have transcription, translation, regulation, and protein activity, and they all start as a stretch of DNA, and they are encoded, post-translationally modified. Localization of the protein and activity and the consequences of it is a phenotype. So open reading frame starts with ATG, if you're looking at the level of DNA, and then it has continuous stretches of codons and has one of the stop codons, which would be TGA, TAA, or TAG. We don't have to really mug up all the codons except for start codon and stop codons. At least these we should have learned by heart. See you in another talk. Thank you very much.